Hi everyone, I am Dr. Shagirtha, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. I am going to talk about isoenzymes. The human body is composed of different types of cells, tissues, and other complex organs. For efficient functioning, our body releases some chemical to accelerate the biochemical process such as respiration, digestion, excretion, and a few other metabolic activities to sustain a healthy life. Hence, enzymes are pivotal in all living entities which govern all the biological process. First, we discussed about what are enzymes. Enzymes can be defined as biological polymers that catalyze biochemical reactions. The majority of enzymes are protein with catalytic capability crucial to perform a different process. Metabolic process and other chemical reaction in the cell are carried out by set of enzymes that are necessary to sustain the life. The initial stages of metabolic process depend upon the enzymes which react with a molecule and it is called the substrate. Enzymes converts the substrate into other distinct molecules which is known as product. The regulation of enzymes has been a key element in clinical diagnosis because of their role in maintaining life process. The macromolecular components of all enzymes consist of proteins except in the class of RNA catalyzed called ribozymes. The word ribozymes is derived from the ribonucleic acid enzymes. Many ribozymes are molecules of ribonucleic acid which catalyze reaction in one of their own bonds and among other RNAs. Enzymes are found in all the tissues and fluids of the body. Catalysis of all reaction taking place in metabolic pathways is carried out by intracellular enzymes. The enzymes in the plasma membrane govern the catalysis in the cells as response to cellular signals and enzymes in the circulatory system regulate the clotting of blood. Most of the critical life process are established on the functions of enzymes. Now we discussed about the functions of enzymes. Enzymes help in signal transduction. The most common enzyme used in the process include protein kinase that catalyze the phosphorylation of proteins. They break down the larger molecule into smaller substance that can be easily absorbed by the body. Enzymes help in generating energy in the body. ATP synthase is the enzyme involved in the synthesis of energy. Enzymes are responsible for the movement of ions across the plasma membrane. Enzymes perform a number of biochemical reactions including oxidation, reduction, hydrolysis, etc. to eliminate the non-nutritive substance from the body. They function to recognize the internal structure of the cell to regulate the cellular activities. With this few introduction, we are going to discuss about the isoenzymes. Isoenzymes are multiple form of enzymes catalyzing the same reaction is known as isoenzymes. They are different in their physical and chemical properties which include the structures. The isoenzyme synthesis from different genes, example malate dehydrogenase of cytosol is different from that found in mitochondria. Oligomeric enzyme consisting of more than one type of subunit, example lactate dehydrogenase and creatine phosphokinase. And enzymes may be active as monomer or oligomer, example glutamate dehydrogenase. In glycoproteins, enzymes different in carbohydrate content may be responsible for isoenzyme, example alkaline phosphatase. First, we discussed about the isoenzymes of lactate dehydrogenase. Lactate dehydrogenase, systemic name is L-lactate NAD oxidoreductase. It catalyzes the interconversion of lactate to pyruvate. So, it's convert the lactic acid into pyruvic acid. LDH, the lactate dehydrogenase, has 5 distinct isoenzymes: LDH1, LDH2, 3, 4 and 5. They can be separated by electrophoresis. LDH1 is more positive charge and fastest in electrophoretic mobility, while LDH5 is the slowest one. The structure of LDH isoenzymes. LDH is an oligomeric enzyme made up of 4 polypeptide chain and 2 types of subunits, namely M for muscle and H for heart, produced by the different genes. M subunit is a basic while H subunit is acidic. The isoenzyme contain either one or both the subunit, giving LDH1 to LDH5. The character features of LDH isoenzymes are: the LDH1 is predominantly found in heart muscles and is inhibited by pyruvate. Hence, pyruvate is not converted to lactate in cardiac muscle but is converted to acetyl-CoA or enters into citric acid cycle. LDH5 is mostly present in skeletal muscle and liver. The enzyme pyruvate is minimal, hence pyruvate is converted into lactate. LDH5 has low Km value while LDH1 has high Km value. For pyruvate, the differential catalytic activity of LDH1 and LDH5 in heart and skeletal muscle respectively well suited for aerobic and anaerobic conditions of tissues. The next one, the electrophoresis of LDH. The isoenzymes of LDH have immense value in the diagnosis of heart and liver disease mainly. In healthy individuals, the activity of LDH2 is higher when compared to other LDH. In myocardial infarction, the LDH1 is more actively increased compared to LDH2, 3, 4 and 5. In liver disease, the LDH5 is increased compared to other isoenzymes. LDH activity in the orbis is 80 to 100 times more than that of serum. Hence, the estimation of LDH or its isoenzyme serum should be totally free from hemolysis or else false positive result will be obtained. Next isoenzymes is creatine phosphokinase. Creatine kinase or creatine phosphokinase catalyze the interconversion of phosphocreatine to creatine. In health, CPK exists in 3 isoenzymes. Each isoenzyme is a dimer composed of 2 subunits, muscles and brain or both. It has 3 different types: CPK1, CPK2 and CPK3. In healthy individuals, the isoenzyme CPK2 is almost undetectable in serum with less than 2% of total CPK. After the myocardial infarction, within the first 6 to 18 hours, the CPK2 is increased in serum to a high as 20%. CPK2 isoenzyme is not elevated in skeletal muscles disorder. Therefore, the estimation of enzyme CPK2 is the earliest reliable indication of myocardial infarction. Next isoenzymes are alkaline phosphatase. As many as 6 isoenzymes or alkaline phosphatase have been identified. ALP is the monomer. The isoenzymes are due to the difference in the carbohydrate content. Most important ALP isoenzymes are alpha 1 ALP, alpha 2 ALP, alpha 2 heat label ALP, alpha 2 heat stable ALP, pre-beta ALP and gamma ALP. Increase in alpha 2 heat label ALP suggest hepatitis, where pre-beta ALP indicate the bone diseases. Last isoenzymes is alcohol dehydrogenase. Alcohol dehydrogenase has two heterodimer isoenzymes. Among the white American and Europeans, alpha-beta 1 isoenzyme is predominant, where in Japanese and Chinese, alpha-beta 2 is mostly present. The isomer alpha-beta 2 more rapidly converts alcohol to acetaldehyde. Accumulation of acetaldehyde is associated with some diseases and facial flushing among Oriental, which is not commonly seen in whites. It is believed that Japanese and Chinese have increased sensitivity to alcohol due to the presence of alpha-beta 2 isoenzymes of ADH. Next, we are going to discuss about the enzyme pattern in disease. For the right diagnosis of particular disease, it is always better to estimate the few serum enzymes instead of single enzymes. Examples of enzyme pattern in important disease are discussed here. Enzymes in myocardial infarction and enzymes in liver disease. First, the enzymes in myocardial infarctions. Mainly the creatine phosphokinase. Enzymes in myocardial infarction mainly the creatine phosphokinase and SGOT, aspartate transaminase or SGOT and lactate dehydrogenase are important for the diagnosis of myocardial infarction. The elevation of these enzymes in serum in relation to hours or days of myocardial infarction is represented in this graph. So first, the creatine phosphokinase or CK, creatine kinase, is the first enzyme to be released in circulation within 6 to 18 hours after the infarction. Therefore, CPK estimation is highly useful for the early diagnosis of myocardial infarction. This enzyme reaches peak value within 24 to 30 hours after return to normal level second or third day. Next enzyme is aspartate transaminase or SGOT. It rises sharply after CPK and reaches peak value within 48 hours and return normal in 4 to 5 days. Next enzyme is lactate dehydrogenase. It generally rises from the second day after infarction, attain a peak value third or fourth day and takes about 10 to 15 days to reach normal. Thus, LDH is the last enzyme to rise and also the last enzyme to return normal level in myocardial infarction. Non-enzymatic biomarker in myocardial infarction is cardiotropin. Although not enzymes, the protein cardiotropin are highly useful in early diagnosis of myocardial infarction. Among these, tropin I and tropin T are important. Cardiotropin I is released into the circulation within 4 hours after the onset of myocardial infarction, reaches the peak value by 12 to 24 hours and remain elevated for about a week. A protein myoglobin is also an early marker for the diagnosis of MI. However, it is not specific to cardiac disease. High serum concentration of brain peptide and artificial arterial peptide are marker for cardiac failure. So next, we discussed about the enzymes in liver disease. Some enzymes, when elevated in serum, are useful for the diagnosis of liver dysfunction due to viral hepatitis, toxic hepatitis, cirrhosis and hepatic necrosis. Different types of hepatitis affect our liver. mainly viral hepatitis, mainly hepatitis B, hepatitis A and hepatitis C. It is a very dangerous hepatitis affecting in the liver. So we are going to diagnose some enzymes or mainly alanine transaminase, aspartate transaminate and lactate dehydrogenase, alkaline phosphatase and 5' nucleotidase. Serum gamma glutamyl transpeptidase is useful in diagnosis of alcoholic liver diseases. Thank you.